So what I would like to share this morning is a little bit an overview of how we see research in scleroderma, how we are progressing or not, and few updates on what's going on at Cedars-Sinai and the Autoimmunity Institute. I forgot to mention that obviously this is a partnership, this event between the Scleroderma Foundation of California, the Division of Rheumatology, the Autoimmunity Institute, and I hope through the talk you appreciate how really it takes a village to take care of scleroderma patient and also to try to advance our knowledge and our ability to deliver the best care. So the first question, that I think is important when we think in terms of research in scleroderma is why do we need to invest more on research in scleroderma? Well, the first brief answer is we don't have a cure yet. And I want to give you a couple examples to show you how this is an unmet need. Skin disease, right? So many of you experience this kind of important and sometimes very tough process of having hardening of the skin. You can see, on the upper left is the early stages. If you remember, those of you that started with puffy hands and little clumsy hands, and then progress into having tightening, that affect the arm. The early phase, we call it, it's edema, puffy, it's clumsy hand, but then it turns into the dryness, the slow progressive dryness and tightening. And then on the later stages, not in everybody, but there is these contractures that happen, change on the texture of the skin, and also other type of consequences of hardening of the skin. So very important problem. How are we doing in terms of treatment? Well, again, when we don't have a cure and we don't have great treatment. So definitely an area for us to invest. As you probably well know, the drug that most commonly is used to treat hard skin, tightening of the skin is mycophenolate or the Cellcept. There are other drugs that were used in the past that are less frequently recommended by experts, particularly in the United States. Some people are referred to the hematopoietic stem cell transplantation. We're going to quickly touch upon that. And then we do use intravenous immunoglobulin as another way to try to tackle skin disease. And the idea is, there is some inflammatory cells. This is a skin, this is a biopsy of the skin, and you see there are these kind of black dots. These are all immune cells, T cells or B cells that are into the skin, and then they are the one that sort of drive the process, make the cells or make collagen excited and increase the tightness. Now, how do we measure that? So those of you who are my patients, when you come to clinic, I start to pinch you. That's what I do for a living. Pinching the skin is a way we have to try to get a sense how severe the skin tightening is. And you see we sort of score how tight that is, thinking that we can really understand what's underneath. And then there are different body areas. I start from the top and then we go all the way down. And overall, there are 17 different body areas with score 0 to 3 and a maximum score of 51. Why am I telling you this? Because this is one of the challenges that we have in our clinical trials. That for a long time we used this as our measure how well we're doing. Is the patient responding to treatment? But you can understand how ballpark this is. It's not perfect. Everybody does it a little differently. If you are swell up because you had a nice pizza or fried chicken, you may be more swollen and the skin is tighter. So it's not a perfect measure, but it was the best we got. And how did we do? Well, this slide, it sort of represents the conventional treatment for skin disease, right? So the methotrexate, mycophenolate, cyclophosphamide, or no treatment. Not all the patients receive medication. And so this was a consortium in Europe, 50 centers, who said, okay, let's put patients in one of these 4 categories. Let's try to treat them and see how well their skin do. So I want you to focus on, so this is the skin score. I told you, the number is 51 is the maximum. Here they say, okay, if this is the baseline, how much over time when you use this treatment do they lose in terms of skin score? Right? So this is a baseline, and then you go down, and as you go lower means improvement. Now, when you don't need to be a scientist to get a sense of, well, if this is no treatment, and these are the treatment, over a year, this is what they recorded. So even no treatment, there was a little bit of improvement over a year. And when you start treating, this is all comers, of course, and among all the patients, there are some who improve better and some who improve less, right? Not every patient is the same. One important concept in clinical care and science in scleroderma is every patient is unique and they have their own way of reacting to drugs. But overall, you know, if you have a very effective drug, you would expect this number to drop much. So they drop 2 points, 3 points, 4 points. How much is 4 points? If out of 51, 4 points is not like a dramatic response. It's a little bit of response over a year. So I wanted to give you the sense that conventional therapy is not working on everybody, and in aggregate, overall, doesn't give us great results for skin disease. We did trials. 2020 was a year where there were so many trials for skin disease in scleroderma, and you know, don't need to learn the name, and don't need to look all these pictures, but I wanted you to reflect on these things. Take for example, the tocilizumab trial. Right? So these are two lines, the red and the blue. The blue is the placebo, meaning the patients did not get any drug, and the red is the patients who got the medication. Again, you don't need to be a scientist. What you see is that going down means improvement. Both improve somehow, and even if the drug arm did a little better, but the difference was not that big. And it's similar all across the board, meaning that all this big investment that the industry did, and we did, and you did, didn't really brought out a convincing one drug that really changed the way we treat skin disease. How did we do on lung disease? Well, it's interesting here. We got a little more updates. So how do we treat lung disease? Again, normally, the initial consensus is to start patient again on mycophenolate, less frequently on these other drugs. Probably we don't use them more, but there were the trials. Don't need to get into the nitty-gritty of the trials, but that show that at least in a subset of patients, the vast majority of patients, there is some benefit of starting them on mycophenolate. And then more recently, over the past few years, there have been more targeted therapy for lung disease. Some of you are familiar with the nintedanib, the brand name is OFEV, the tocilizumab, and more recently also the rituximab is a B-cell depletion therapy. So these are considered more targeted, and somehow the FDA approved them because they were giving some benefit. And I'm gonna go back to them in a moment. And then obviously we still have the possibility of doing stem cell transplant, the old-fashioned stem cell transplant. And this is an area where also we may have new approaches, and we're gonna talk about some new exciting therapy. Don't forget lung transplant. We are lucky that at Cedars-Sinai we have a very strong lung transplant program, which in some patients where we exhausted the ability to use medication, it's actually a life-saving way of treating lung disease. But how did well perform these new drugs? This is an example, is the SENSIC trial. So this is the drug is nintedanib. And I wanted you to reflect that the FDA approved it because what it shows, so in this case, going down is worsening, is not improving, right? So this is lung function at baseline and then over a year, and you look what happened over time. And basically what the trial showed here was that the drug slowed down the worsening as opposed to the placebo. Placebo meaning getting the sham treatment. So yeah, the FDA approved it. Certainly we got some benefit, but still patients are losing function. So what I'm trying to emphasize is even if it's an improvement of what we had before, still is not on target. And even on this trial, there is like a 30% of patients who do not respond at all to the therapy. To say that even where we had some progress, still there is a lot of work to do. How about the stem cell transplant? You know, some of you are familiar with that. You know, this is like a very, at some point we felt that aggressive immunosuppression is the way to go. So not just a little bit of a pill, but doing like a full-blown, almost chemotherapy level intervention. You know, the drug used for the stem cell transplant is cyclophosphamide. And you really wipe out all the immune system, and then you use the stem cell to get the immune system back, hoping that the immune system, the new immune system will behave and the disease calm down. So there were some sense of that this was more effective. So we know that aggressive immunosuppression or more assertive immunosuppression works. The table here is just to show you that in terms of, this is the skin score, the patient on the stem cell transplant dropped 20 points, which is much more than what I showed you before. Before the conventional therapy, they were dropping 3-4 points. This was much more effective compared to the conventional therapy. And even the lung function here improved, but there was a price to pay. And the major emphasis here is this red circle, is that it was such an aggressive treatment that there was high treatment-related mortality, meaning you have to tell your patients, yeah, it's an effective treatment, but 3 to 10% of people die because of the chemotherapy, not because of the disease. And so you understand this is a risk that has to be discussed, but that was not the only problem with this approach. The biggest problem, it was that we started to see flares, meaning that the hope that by doing this one time, you were able to wipe out the disease didn't pan out, and particularly those who had severe disease to begin with, variable time after the stem cell transplant, the disease was coming back, and sometimes even coming back with a vengeance. So we knew that this is an option, but it was not the answer, because there is mortality, there is also the morbidity, meaning people also can get infections, and then there is this long-term fact that this is not a one-time done. Oftentimes you have to resume therapy anyway afterwards. So not a solution, right? Now, I think I convinced you why we need research, because we don't have a cure yet. Then there is another layer of complexity on our patient population that, for scleroderma, one size does not fit all. Every patient is unique. We cannot go by the book and say, oh, you have scleroderma, this is the treatment plan for everybody. We have the fact that the disease is unique in every patient, and the biology of the disease change over time. I always use this slide. These were, patients of long time ago, but every phase is different, meaning everybody experienced the disease in a different way, and even the way it impacts the body is different. And think of the complexity. This is not a one-organ disease, right? So if you have autoimmune thyroid disease, you have to deal with the thyroid. That's pretty much about it. Get the hormone replacement, and you're done. Scleroderma affects any organ, and so it's almost like a puzzle. Every patient have their own unique combination of challenges, and so this certainly poses a challenge for us to find a cure, to find better ways of treating the disease. Another reason to invest on research is really how do we learn how to assess for these different organ manifestations, and when you think about new novel treatment or novel diagnostic, how do you take into consideration this complexity? So what are we trying to do also is how can we face this? So, there is an early stage of scleroderma. There is an early stage of scleroderma where mostly, and some of you remember, started with the Raynaud's phenomenon, the cold sensitivity, and then the doctor found that there was a positive antibody, and then say, oh, well, you may have scleroderma. And we know there is this early phase where basically it seems that nothing else is going on, but by the time you have established disease, and we start to, for example, seeing the skin tightening and everything, we know that internally the organs start already to be affected. So one challenge in research is really, can we get to an early stage to study the disease and to try to treat the disease more effectively? Because if you wait too long, then it's much more challenging to be effective, to reverse damage that already happened. So one area that in research we're trying to get organized, and I think the value of having dedicated center is really get the patient early to experts who can really start working during this early stage when there is minimum damage, but we know what to expect. We know we have ways to start looking carefully inside, even if there is no apparent damage or ongoing process. We can really, having this large cohort, start to study the disease early on. And that will give us an opportunity to say, can we reverse the early process? Why is that important? Well, think of it. Scleroderma has different souls, I always said. There is always an autoimmunity. The immune system is acting up, is damaging the body. But then there is the fibrosis, the scarring, excessive amount of scarring that some patients experience in a particular dramatic way. And then there is the blood vessel damage, the vascular injury, which is very common pretty much on everybody, the Raynaud's and some degree of damage of the blood vessels. These three components, though, they're not always the same. So what we learn by studying the immune system and all the biology of the disease is that during early phases, it's more likely that the activation of the immune system is important. The inflammation, all these immune cells that are causing the attack. But over time, if you look at the wheel here, gets smaller and smaller. In other words, as the disease progresses on the long term, it's not that there's always the same degree of immune system. Then other processes take over, like later on the scarring becomes more important. And then on the long run, the blood vessel kind of damage, it becomes more dominant. So when you think in terms of studying, doing research on what's the best treatment for scleroderma, you can't really think of only one drug. So early on, sure, the immunosuppression, so most of the drug I show you were basically drugs that were designed to impact the immune system activation. It makes sense maybe during the early phases. But as you move forward, you have to think different. You have to sort of start to say, do we have antifibrotics, meaning drugs that can really impact fibrosis directly to reverse the fibrosis? Do we have drugs that protect the blood vessel? And so what I think is important for us when we do research in scleroderma is to thinking outside the box and say, okay, we don't have to focus only on immune suppression. We need to start thinking, how do we change instead the other type of the other biology of the disease? How do we impact fibrosis? How do we protect the blood vessels? So some of the research that, particularly here at Cedars and the Autoimmunity Institute, we're doing is really focused not only to study the immune cells, but also looking at the fibrotic process, and I'm sorry that Dr. Bottini is not here. You would have give you a good example how they are studying the fibroblasts, which are the cells that makes collagen. And we are also starting a program where we're going to look at the blood vessels and how this lining of the blood vessel gets sick early on and then cause the blood vessel to get damaged and driving then the late stage of the disease. So these are really the challenges that I think are important to consider when you think in how do we study the disease, how do we invest in research. And what is very clear to me is that this has to be a global effort with all the community being involved. You need centers that have large cohort of patients that are participating into this research effort because, again, this is a rare disease. It's not that there are millions of people that have scleroderma. So you really need to put together the resources and try to really start answering this important question. Now, another important unmet need or challenge that we have on research is this. The disease burden is significant beyond skin and lung. So when you look what kind of trials are being done, research trials are done in scleroderma, most of them are lung disease, important, don't get me wrong. I'm not saying that they are not important. Lung disease, we were doing skin disease, now much less. There were some trials on Raynaud's, and now they're not investing much on that. There are very few. But the disease is much more than the lung, the skin. We know, think about the vascular manifestation. Some people experience, ulcers, infarction of their digits. There's not much research going on this. The GI tract, we're gonna hear from Dr. Rezai today. This is 95% of patients with scleroderma have GI problems. Did you ever heard about a clinical trial on scleroderma patient with the GI motility? There are not many. There are trials on IBS or general GI problem, but look, I don't want to get into the detail of the slide. This is to show you that from the top to bottom of the GI tract, there are serious problems, starting with, teeth loosening, difficulty with chewing, the esophagus gets strictures or like the stomach start to bleed. The small intestine, you got bacterial overgrowth and then lose the motility. All the way, even anorectal sphincter incontinence. So you name it, unmet need. There's not much research going on this. And then think about another problem that some of you are very familiar with, calcinosis. How many of you, my patient ask me, what can I do about this calcium deposit? So not everybody has this, but for some reason there are some patients that are more likely than others to build up this big calcium deposit that sometimes, can become very problematic. and it can affect large joints. This is a wrist. This is the X-rays of this wrist. Look how much dense calcium you have here. A lot of disability, but yet there's no trial. There is no investment on this. So this is to show that the disease has a burden that is beyond just the main organ, and yet we have to have an investment that start to address this important determinant of a quality of life of our patients. And then don't forget, we are not just cells and blood and biology. We are more. We are mind and soul. And I think it's important that we appreciate that having scleroderma, a chronic disease, it's more than just having tight skin. Look at all these experiences that the patient can have. So affecting the physical, mental, emotional, social, financial, and spiritual domain of the life of a person. And so how much research is going on these things? What are we doing to address also these important factors that determine ultimately how well you're doing? We look at, I can pinch you and I can say, oh, the skin is getting better, but maybe you don't feel good. You don't feel great, and so when we think in terms of our scleroderma program, I truly, sincerely hope that as we grow our cohort and we get more resources, we can start to tackle also at this level ways to improve the quality of life and the well-being of our patients. And then finally, just to talk about why we need more investment on research, is that drug development is very slow. I use this slide to give you some sense. So imagine that you start from research and you do microscope and then test on animals, and then finally you bring to clinical trial, and then you submit to the regulatory agencies for approval. The whole process, from the moment you say, I have a molecule or a target that can become a great drug, from that moment, if you're lucky, it's basically 10 years before it gets to fruition. Okay, so that's the reality. And don't forget the cost. On average, to get one drug from here to here, the average price for developing is 1 to 3 billion. So what does that mean? Well, if you have high blood pressure and you're trying to develop a drug for high blood pressure, there are so many people that a pharma company say, oh, sure, if I get a new drug there, I have a huge market. But for scleroderma, it's not that we have a lot of patients, so it's not always easy to get company to invest and say, oh, I put $3 billion for something that serves a smaller community. But fortunately, because it's recognized as an orphan drug disease, there is still willingness to invest on this disease. But just so you're aware, it takes a long time and a lot of investment to do that. Now, how do we respond to these unmet needs? So what do we do? What has been happening in the community in research that can really start to answer to this challenging situation, right? So the first is that I call it the power of observation. Precisely because every patient is unique, precisely because when we do clinical trial, we know that at best there is a certain number of patients who respond, but there is always somebody who do not. We need to pay more attention to our patients. We know, and again, goes to the concept of having centers who are really dedicated to scleroderma patients and can really look at the patients in aggregate or to the individual patients, observing what's unique about the disease process in this patient. And that's critical to generate good idea. And that's why I think cohort or registry are critical to really have better sense of how to address the disease, how to approach, where to start, what groups or what subset of disease should be tackled. We definitely are fostering a faster connection. I call it bench to bedside and back. I'm going to give you an example of that. Leveraging the new technology. It's a fast evolving field of having technology at our fingertip, and we should really use it. So I give you this concept of the forward and reverse translation in scleroderma research. So the bench to bedside is this way, right? So you study something in the lab, and then you try to bring in to the patients, in clinical trial or cohort. But I think what's important now is if you have the right setting, is also you go the other way around, that you don't just start doing basic science and then eventually it's going to get to the patient. But by doing a good job observing the disease, studying the disease, see what previous treatment have done, what kind of, you are able to really then go back and tell the smart guy in the lab saying, hey, I think there is something about this. Can you study it more deeply? Now we can do this very effectively, very efficiently, and certainly one of the reasons I came to Cedars-Sinai is really to build an environment that allow to have this process. And I give you, the framework of how the scleroderma program at the Cowden Immunity Institute is really approaching the discovery with the goal to identify new target for treatment, developing new treatment possibly, and improving management of scleroderma. So, we have, fortunately, people and an infrastructure that can really study the different organs at the global level and also with the tissues that we have available. Then you can sort of assess whether something is working on a preclinical level. So we can do we have a machine that can do high throughput analysis on the cells, and then we can see how those change their behavior. And we also have a very important area of animal models that are available to test in a preclinical setting the efficacy of these drugs. And then you bring it to the clinical side. So here, we have clinical trials that some of them are from industry, some we hope to be able to do our own investigator-initiating clinical trial to test this drug. And you have the cohorts, meaning that, don't get me wrong, the fact that our patients are allowed to participate into our prospective cohort allow us to study their clinical manifestation over time. And that actually, since we are treating them, give us idea of what's working or what's not working. And so you really are able then to define what the targets are. Are there something more efficacious than not? And Dr. Dan First always said that there is not a bad trial. There is not an unsuccessful trial. Every trial teaches us something. Even the trials that fail, we can learn something from those. And that's what I mean. If you have the right setting, you can learn what works and what does not work. You can say, oh, maybe there is a group of patients, not everybody, but that is more likely to respond to this treatment, and then bring it back. Bring it back again, the idea of the circle that is not only one way, but you can definitely, from the clinical side, tell the smart people, tell the colleagues that are working in the lab, say, hey, we should invest in this direction. And I'll give you an example of that. So for example, scleroderma, we can do a clinical classification. As you probably know, most of the people are divided into limited, diffuse, or they have overlap features. But what we learn is that even within a given category, it's not that everybody is the same. You see different colors. There is a variability of people. And so having cutting-edge technology, and for sake of time, I won't go into what kind of powerful tools we have. We are able to do what we call cellular and molecular profiling. So we got samples from the patients, and then we can sort of look at, at the level of the DNA, of the cells, at the level of the tissues, how these different patients are unique. And then sort of come up and say, now we do this profiling. Within this group, there are the blues, there are the red, there are the yellow, and there are the green. And those doesn't matter if you have limited, diffuse, or an overlap. What matters is we're finding something particular and unique in your DNA or in your cells that set you apart from other people and put you in a category that is unique. And so what do you do? Well, then we call what we call endotype, meaning instead of saying, let's classify people because from the clinical standpoint, what if we use all this data that we have in science and classify from the molecular standpoint, meaning it doesn't matter if you have diffuse disease or limited disease on your skin. What matters is you have the yellow signature. Your immune cells are behaving like all the yellows do, and it's different how the blue behave and so forth. And so what's the outcome? Well, then the treatment is tailored, is targeted. So this is the concept of personalized medicine. So all the trial that we did certainly pointed to this direction, that one of the reasons they fail, I show you the trial that didn't work, is because even if you say, oh, we're going to treat lung disease, and it's not that everybody has the same process. If you treat skin disease, not everybody has the same type of cells or they don't behave the same. And it's a little bit what happened with cancer, right? Nowadays, you don't treat necessarily prostate cancer, breast cancer. They look into the cancer, look at the genetics, and then doesn't matter what cancer, there are targeted treatment that goes to the specific molecular biology of this cancer. So we hope, and this is one highlight, one important area that we are investing here at the Cloud Immunity Institute, is really to say, can we figure out what's the code of the unique patients? And therefore, we can even use better the treatment that we already have. Doesn't have to be, inventing new things. But like the treatment that we have, maybe we are able to predict who is going to respond to drug A, who is going to respond to, to drug, the blue one or the green one. And so one is to use better the drug that we already have, and the other is then to have a process to discover new drugs. So I hope this is clear, but this is an important area when you think in terms of what's needed and what kind of new research we are doing. That's what we're doing, that's what we're thinking and aiming to. Now, we're also doing better on a clinical trial, so I won't spend time, but basically, you're not going to see true placebo trial anymore. So we allow background therapy into the new trials, which is important because at least we don't lose much ground, and we allow patients to have some treatment anyway on the background. We are stratifying patients, so we're learning that being good at defining the autoantibody or certain biomarkers allow us to start clustering patients that are more similar and more likely to respond to therapy. We are getting together as a community to have more uniform protocol. So one of the problems is every trial was slightly different, and it was very difficult to compare them. So I think now, because particularly the centers that have dedicated scleroderma program are getting together on these trials, we're trying to sort of share uniform protocol, and we are trying to define new ways to determine that if the drug is working or not. Again, all days we were just pinching. Now we have better score that hopefully will give us a better, more objective measurement how well the patient is responding or not. And it will be also a help with us to get the drug more approved, to add more tools in our repertoire. And then we will talk about design of the trials. This is an example, for example, is the platform design that the Conquest trial, and I'll talk about that in a moment. And then testing new therapy, and we will speak about CAR T-cell therapy as a new exciting area of thinking how to more effectively treat scleroderma. So what are we doing at Cedars-Sinai? So everything starts with excellence in clinical care. There is no doubt about it. We are investing on creating this large prospective cohort, and I hope I gave you enough justification of why it's important to have a large number of patients to follow over time. And I have to say, in our discipline, we're lucky. I don't recall a single patient who say, I don't want to be part of research, or I don't want to be a guinea pig. Everybody that I work with enthusiastically say, tell me what I can do. I know it may not help me today, but may help people down the way. So I think this partnership is critical. We created, so not only a large cohort that we can follow over time, an important biorepository, integrating it with all the research laboratory. So you have ability to look at the disease, how the clinical variable change over time, and then to match that to the biology, how the cells change their behavior. Recruiting, we try to recruit smart people. I'm going to show you some, and then, investing in the most exciting new clinical trial. And I think, the possibility, one of the uniqueness of Cedars-Sinai, besides developing all the clinical program, was also to have, an institute dedicated to autoimmunity, with scleroderma being a flagship program. The cohort now that we have here is more than 300 patients, and everything starts from the care. These are the people that you may recognize if you come to Cedars. We want to start from here. We want to be sure that the patients before, during, and after the visit have what it takes to really be taken care of. We want to know what's unique about every patient, and to be sure that we are trying to support them over time. We are getting patients from all over. That's a good sign. It's really rewarding knowing that. And I always, people ask me, do you have trouble getting patients to your clinic? I say, no. I think scleroderma patients go through a lot of stages where they don't find the right person that knows about the disease enough, and they are the most invested in looking at a place where they can get help. So fortunately, LA, we have at least 2 big programs in scleroderma where you can get the best care, but there are other around the countries. Bottom line, people know that there are places where they can get the comprehensive care, not just the scleroderma doctor, but also the team of people. And today we're going to hear from Dr. Zaman, that is our pulmonologist, Dr. Rizai, gastroenterologist. So a team of people, a multidisciplinary approach that really allow us to take care of all the different diseases. And then I talk about the Cow Immunity Institute. There is the technology that we're developing and we're investing on. We are purchasing technology, we are purchasing knowledge, we are recruiting very good people to really deliver this full circle that goes from best care to study the disease and then bring it back to the clinic. And this is an example just to say how it really takes a village. In clinic, we have a dedicated team of people that do the consent, the blood draws, that help me with the database to get all the information that we need from a clinical standpoint. And they are really critical for us to be able to create this cohort. And then the KAI technology unit with Shannon Madison, they make sure that if you give your sample, your blood or your skin, we don't waste it. They are phenomenal at processing the samples, creating everything, setting up the stage for the investigator to be able to study this at the best level. And these are some of the investigators that we, when I say smart people, I mean these people here. So unfortunately, Dr. Bottini is not here, as Tina was saying, has some kids that are sick. But I hope there will be new opportunities. He's the director of the Cow Immunity Institute, and he is devoting a substantial amount of effort to really study scleroderma. But we also have Richard Ainsworth. He's a computational biologist. So nowadays there are so many data. You need somebody that can crunch number in a fast way, not with a piece of paper, but with a supercomputer. And so we recruited him to really help us being able to accelerate the process of learning all the biological data. Chloe Choi, she's a lung biologist that came from Cleveland Clinic, and we hope that she will help us studying the biology of lung disease in scleroderma in particular. And so she's very eager to participate in this project. And even we attracted interest from abroad. So Laura Polivka, she's an MD PhD from Paris, who will be visiting for us for 3 years to study the scleroderma in kids. So she has an interest on the genes that may ignite scleroderma, may drive scleroderma in kids. And so she selected to work with us, and particularly with Dr. Bottini. And so I think this is really a testament to how you can create a place where people gather to really study the disease very deeply. To conclude, and I know I'm going a little longer, but just quickly, 3 examples of clinical trials that we're doing here. So the first is the Athena trial. So this is a classic trial. It's done to basically answer the question whether this drug, I don't even pronounce the name, is effective in treating scleroderma lung disease. These are patients who have disease for less than 5 years or less. They need to have diffuse skin disease, and they need to have proven interstitial lung disease based on CAT scan. And so this is a year-long trial. It's an example to say that it's not limiting patients who already are taking treatment. So all days we were excluding patients who were already on some medication. These new trials don't exclude that, which I think is a plus. So we can have stable therapy and then add on top of that the new treatment and then assess how patients do over time. So this is an active ongoing treatment that we are enrolling. An upcoming trial is called the Conquest trial, also geared towards patients with interstitial lung disease. The uniqueness of this is, number one, the sponsor is the Scleroderma Research Foundation. So not a pharma, but a foundation that is dedicated to scleroderma, with the idea that they really have your best interest at heart, but they also think outside the box. They created something unique that never happened before, creating a platform trial. What is a platform trial? Basically, is a perpetual platform where if there is a new drug, you can enter into the trial, And you can have more than one trial. At the moment, they have two therapies, therapy A and therapy B, but potentially they can add a therapy C, and then have a placebo arm that serves for all these different arms. So in other words, in the conventional treatment, you're randomized, either you get the drug or you get the placebo, right? It's a 1-on-1. The platform trial, because it leveraged the fact that there are multiple treatment and one placebo group, basically you're randomized 2-to-1 or 3-to-1. So you have even more chances to be on some of the drugs, and there is a group that will serve as a comparator for all the different drugs. So it's very exciting because it maximizes resources and maximize likelihood of the patients to also receive some treatment as they go through the trial process. And again, inclusion is similar. One limitation is all this trial, want to tackle early disease, so 5 to 6 years from disease onset. That's always the biggest limitation when you have to approach patients and say, hey, can we consider trial or not? But other than that, it's just confirming that there is interstitial lung disease and stable background therapy. And then this is the biggest news, and the new trial we're setting up here at Cedars-Sinai, which is basically a phase 1 and 2. Phase 1 and 2 meaning first is safety and some efficacy, meaning we want to be sure that the patient can tolerate the drug, that the side effects are acceptable, and then start to see if there is any sign of efficacy on the biology of the disease. If this works, then it's brought to a phase 2 or phase 3 trial where really then you make the case that the drug works and it's the future of how you treat this disease. But what is this treatment? It's called CAR T-cell therapy, chimeric antigen receptor T cells directed against CD19 cells, is B cells. So the interesting part is that this is geared towards either active skin disease or active lung disease. So both can be accepted, and the treatment basically came into the news last couple of years ago, where we started to have news release about lupus patients treated with this CAR T-cell therapy with remarkable results. The doctor was saying we throw away the medications, and what is it? Well, CAR T-cell, it's an engineered cell. So in other words, this is a T-cell, so your immune cells. And basically, the process is to take them out from the patient and engineer them to express this antigen receptor. This kind of red part is something that we decide what it is. And you use a sort of a virus transfection to make the cells express this red kind of fragment. And this red fragment, imagine, is like basically a receptor that is specific to something that you want to target. In fact, these have been developed in cancer at the beginning. So we're getting this from cancer. So imagine that you have this kind of red fragment that really recognizes exactly what the cancer cell has on the surface. And by engineering, you directing your immune system to really target the cancer cell. So then these can bind, and then they kill the cancer cells. So what if we do this for autoimmunity? So in other ways, we know that there are some bad actors, bad cells that cause the autoimmune disease. Again, I show you the trajectory that early on, certainly the immune activation, the autoimmunity is more vibrant. So what if, instead of a cancer cell, let's say we recognize there are some cells that we think are more problematic and likely the one that drives the disease, and we can do the same thing, get the cells from the patients, your own cells, engineer them, express sort of a magnet or something that can target specifically just the cells that we think are causing autoimmunity, and then we re-inject them to go kill those cells that we don't want to have around. And I think that's what's happening. This is the CAR T-cell therapy concept. And that's the cycle, right? So you basically collect the blood from the patient. You get a lot of T cells, you send them to the lab where they engineer them to express kind of this receptor that you think is important. And then you sort of grow them, you multiply them, so you create a good army of these new engineered cells, and then you re-inject them into the patients. Your own cells, engineer, re-injected to target specific cells. At this stage, the cells we are trying to target is B cells. So it's similar, if you're familiar with rituximab, That's a drug that empty T cells from the body. So they're targeting similar cells, but with some advantages. Advantages, one is that these are your own cells, so differently from a drug, they know where to go. They circulate into tissues more effectively. So we believe that through this therapy, cells will really go into the skin, really go into the lungs more effectively than a monoclonal antibody. Second, they stick around longer. So hypothetically, and again, this has to be done, is a trial that will start. You do one treatment, you don't need to do it every week or every, the data shows that these cells can stick around for months and months, even a year. So if this is working, it may limit the amount of toxicity from other drugs that you're taking. And then third, the T cells are naturally primed to be effective killers. So I think also the degree of killing, the degree of eliminating cells that you don't want, it will be more effective. So very exciting, and I think we are poised to get this started at the end of the year or at the latest at the beginning of the new year. I know there are other centers who also will invest on this. There is not much toxicity in the chemotherapy for cancer. Obviously, there is a cancer to kill, and the cancer, when it's a lot of cells that release a lot of mediators that can cause inflammation and side effects. But because here is autoimmunity, we're not dealing with a bulk of cancer. The process seems to be much more benign and well tolerated. And it has the advantage over the classic stem cell therapy that is not so toxic. And I think, it's really giving us hope that this can really put the disease at least into remission in a more sustained and effective way. So more to come, but just so I'm sure you read about these CAR T cells. I hope I gave you some sense of what these are. So to conclude, I want to say that we don't have a cure, but I definitely want to emphasize that scleroderma is treatable. Okay, we can take care of you. We can definitely address the needs with this multidisciplinary comprehensive effort, and hopefully by partnering up, we can start to move forward and closer to find a true cure for the disease. But definitely more treatment, more effective treatment are needed. Clinical research takes a long time from the way you conceive it to when you bring to approved therapy. I appreciate that, but don't get discouraged, okay? There is many different kind of research that is ongoing at different stages. And I think, participating into these efforts, if anything, will allow us to be more effective in finding a better way to treat the disease. We are in a stage where we can understand better the biology, and again, center like ours, where there is a dedicated institute to study the disease, allow us to understand the biology. But I want you to remember the circle, that is not only that you studied in the lab, but it's also by looking at the patient that you learn what may be the biological underpinning of the disease. I show you that we are making progress in the way we do trial design, and that there are new treatments actually that are really revolutionary, a paradigm shift. Not the classic drug that you inject, but this time we're working with the cells, your own cells, and hopefully be more effective. And ultimately, personalized care. The technology allows us now to look deeper than just pinching your skin and say, oh yeah, you belong to this category. We can really go to the cells or to the molecules that drive the disease and say, okay, you belong to a certain group, and maybe we have a treatment that is more specific for this. Research is critical to achieve real progress, and I want to emphasize that I'm as motivated as you, but I know that this is a partnership, and we are in this together. Thank you. And we will have time.